Hello students, today we will discuss about DNA damage repair. I will discuss about the causes or sources of DNA damage, their effects on single strand as well as in double strand, consequence of the DNA damage, the significance of the DNA damage repair as well as different types including their mode of action. Let's talk about the spontaneous as well as induced causes or sources of DNA damage. As spontaneous causes, the replicational error, base selection error as well as repair error give rise to the spontaneous causes of DNA damage, or the base tautomerization, spontaneous oxidation by endogenous reactive oxygen species give rise to differential kind of the DNA damage. As well as we can find there are several induced causes like physical mutagen, heat, ultraviolet radiation as well as chemical mutagen like 5-bromouracil base analog. There are so many industrial as well as environmental agents like deaminating agent, oxidative agent by creating reactive oxygen species can change the nature of the base and they are called as endogenous sources. Transposon virus, they are the biological mutagens which can cause the DNA damages. The forms after DNA damage may be concentrated at the single strand, so backup is there on other strand, or maybe double stranded damage. Single stranded damage, it includes base modification, it includes mismatches, it includes strand break, loss of base as well as cross-linking. Let's talk about them individually. Base modification may be oxidation giving rise to 8-oxoguanosine. This oxidation may be spontaneous or may be induced by endogenous ROS or exogenous ROS. Alkylation, 7-methylguanosine, 2-methyladenine, O6-methylguanosine, different types of alkylated bases can be happen either by endogenous causes like S-adenosylmethionine or by ethyl methane sulfonate, the bases may be alkylated. Next, hydrolysis. Hydrolysis gives rise to deamination, depurination as well. So, we can find also the bulky adduct formation by the presence of industrial as well as environmental causative agent like polycyclic aromatic hydrocarbon. So, they give rise to benzopyrene DG derivative. So, you can find these all are the modified base, alkylated base as well.
Ma'am, how does an alkylating agent can cause DNA damage?
6-methylguanosine can introduce thiamine. So, you can find the point mutation that is from GC to AT. Otherwise, these mono or dialkylating agent can cross-link the intrastrand or interstrand. Let's talk about the mismatch. So, these mismatch either may be GT, GA, AC or other ionosine related mismatches can be found. So, mismatches are not the unusual DNA damage. They can be found, but they must be repaired before other cell division. Otherwise, it can introduce the point mutation to the next generation. Single strand base loss. So, by the action of glycosylase, a purinic or a pyrimidinic site can be produced as well as intra-strand cross-link that can be mediated via different types of radiation as well. And ionizing radiation can make single-stranded break as well. Let's talk about the double-stranded damages. It includes the distortion in the helix by double-strand break as well as making cross-link. So, this is the picture of break as well as double strand, inter-strand cross-link. The damage may be direct by ultraviolet radiation having very high amount of energy content H-nu. The phosphodiester bond can be broken or the photoadduct can be formed. Photoadduct may be cyclobutane pyrimidine dimer. A cyclobutane ring C double bond C is made in between two pyrimidine, maybe thiamine or thiamine cytosine, okay, or 6,4 photoproduct can also be made. Apart from these, indirect damage can be introduced by ultraviolet radiation via propagation of reacting oxygen species which can oxidize the bases, so can introduce the mutation as well as DNA damage. Let's talk about the consequence of DNA damage. So DNA damage can introduce senescence, aging, necrosis, apoptosis, halt in transcription, halt in replication, DNA aberration, as well as it can introduce repair as well. So cell has protection strategy. What are they? The natural protection means increasing proofreading capacity, that is base selection, fidelity of DNA polymerase. Okay. Another type of natural protection is that the DNA helix is arranged in such a way it does not allow the mutagen to come inside the helix, as well as the repair system maybe excise the damage, maybe tolerate the damage, maybe reverse the damage. Let's talk about the repair. So when I will talk about the repair, I will talk about the history, I will talk about the factors, I will talk about the significance, strategy and different types related to their mode of actions. First, Kellner was observed the UV related photoreactivation in case of E. coli, and you know Kornberg associates the DNA polymerase 1 related to the excision repair. So cell type, cell age and differential environmental condition can affect the repair. So the repair is needed.
Ma'am, if DNA is not repaired, what will happen?
If the DNA repair is impaired, that give rise to necrosis, senescence, the differential rate of evolution as well as tumor leading to the cancer. Let's talk about the differential strategies of repair. Okay, it includes the damage reversal, just the portion earlier stages of the DNA can be reversed back, or removal just by breaking the phosphodiester bond, the damage can be excised out, or tolerance by replicational bypass, translational polymerase or recombinational repair, the damage can be tolerated. Let's talk about the types of repair. Yes?
Ma'am, what is direct reversal of DNA damage repair?
Direct reversal means again it is going back to its original form without breakage of the phosphodiester bond.
Okay.
Very nice question. So these repair can be concentrated only at the single DNA or maybe to the double strand. Okay, single stranded repair is very easier because the backup is there on mother strand. Okay, so let's talk about the single stranded DNA damage repair. It includes the damage reversal. So I will talk about ligase, photolyase as well as alkyl transferase action, as well as damage excision or removal by excision repair as well as mismatch repair. So damage reversal strategy, it includes the ligation by ligase, photoreactivation by photolyase as well as alkyl group transfer by alkyl transferase. Ligase, you know, if very short gap is made in between 3' hydroxyl or 5' phosphodiester bond, so ligase can just join them up if nick is very short. And in case of E. coli, it needs cofactor NAD+. In case of other like T4 and other eukaryotes, it needs the cofactor that is ATP. Photolyase, you know, when the bacteria is exposed under ultraviolet radiation, so by direct damage it can make thiamine-thiamine dimer, maybe cyclobutane pyrimidine dimer, usual CPD, or 6,4 photoproduct. Now after such UV exposure, if we expose the cell under UVA light or normal light having 300 to 500 nanometer wavelength, a surprising thing happens. The damage, the thiamine-thiamine dimer is getting removed, gaining the original form. So that is the damage reversal, gaining original thiamine-thiamine version from the dimer condition. Okay, so that was done by the presence of photolyase. Okay.
Ma'am, can you explain the mode of action of photolyase?
Okay, before going into the mode of action of photolyase, let me talk about their total component. It is a type of flavoprotein, okay, mainly present in E. coli, but there are several other consensus domain found in case of eukaryotes also, like Cry1 and Cry2 domain found in human. In case of Arabidopsis thaliana as well, the cryptochrome that is related to maintain the circadian rhythms also have this sort of domain also. So what is actually their action? Let's talk about their action, but before going into the action, they have two chromophore, light harvesting chromophore. One is constant, that is reduced FADH minus, that is FADH2. Another cofactor is variable, giving rise to the different types of photolyase, maybe folate photolyase or diazaflavine photolyase. So what is folate photolyase? Having pterin methinyl tetrahydrofolate as a differential cofactor. Okay, so that is found in folate photolyase, and another is diazaflavine photolyase having diazariboflavin moiety. So this actually accelerates the light harvestation by FADH minus. This is the main thing. So what is happen? What is the mode of action of photolyase? It first harvests the light energy. So electrons are excited and they transduced from FADH minus to convert it to FADH2. And these excited electrons or high energy is giving the energy to thymine dimer to resolve them up. 6,4 photolyase is present but not found in case of placental mammal. Okay. So we can find the differentiation according to the amino acid sequence as well as in case of prokaryote as well as in case of eukaryote. In gram positive, gram negative as well as halobacterium, halobium, we can find such light harvesting photolyase activity. Okay, their amino acid homologs can be changed. Alkyl transferase, the alkyl group, right, which is made at O6 methyl guanosine, alkylating agent either exogenous or endogenous. They can transduce what? Some sort of point mutation, GT mismatch, you know. So the group must be transferred. Alkyl transferase is found in case of eukaryote like MGMT, O6 methyl guanine methyl transferase. Having a cysteine residue consensus sequence, it accepts methyl group from the damaged O6 methyl guanine. Okay, but the problem is that it is a suicide enzyme. After accepting the methyl group, it is sent for ubiquitination, proteasome mediated destruction. Okay, so that is very wasteful. But this counterpart is also found as OGT protein in case of bacteria, again having one cysteine residue. But in case of bacteria, there are different other alkyl transferase also giving rise to adaptive response like ADA protein. Okay, it has two consensus conserved amino acid cysteine. What are they? Cysteine 69, cysteine 321. Okay, by these cysteines, they can take up the methyl or alkyl group. May specificity be changed, but it can take them up. Okay, after taking up the alkyl group from the damaged DNA, a conformational changes happen within the ADA protein. Okay, so itself after conformational change, it accelerates the gene expression from the promoter region of ADA itself, ALK A, ALK B. There are different other kinds of alkyl transferase as well as 8A and 8B, which are deoxygenases, which can remove the oxidized base. So in this way, they can evoke the oxidative responses, they can evoke the adaptive responses in the cell. So they are not yet wasteful. Let's talk about the damage removal strategy. That is the excision of damage, cleavage of phosphodiester bond. It includes the excision repair as well as mismatch repair. Excision repair, base excision repair either or nucleotide excision repair. Let's talk about the components as well as mode of action of base excision repair. there are so many glycosylases are there, specifically UNG, UDP-N glycosylase, which scans throughout the minor groove of DNA to find any glycosylation is there. Okay, so the cytosine, if it is getting glycosylated, uracil may come. Fine, but uracil is not normal base in DNA. That is the normal base for RNA. So UNG or glycosylase cleaves beta-N glycosidase linkage and makes apurinic or apyrimidinic site. This invites epi-endonuclease to recognize the epi site and make a cut at the 3' and 5' position of the epi site. So glycosylase makes epi site, epi-endonuclease makes a cut at the 5' end. Next, DNA polymerase, especially DNA polymerase 1, replaces such mononucleotide with respect to the mother strand, and it has the lyase activity by which it cleaves the phosphodiester bond and ligase join them up. Sometimes another enzyme, DRPase, phosphodiesterase, individually can take part in such process. So from picture you can find the presence of glycosylase as well as epi-endonuclease. Let's talk about the components in eukaryotes. So that I have talked about the glycosylase, epi-endonuclease and DNA polymerase 1 ligase is needed for prokaryote. The same thing is applicable for eukaryotic system also. Eukaryotic system means like C. elegans, Drosophila melanogaster, like Leishmania species as well as in mammals. If the base is getting damaged, so the short repair, the short patch, the short base is getting repaired in case of eukaryote. Either by short repair is possible or long patch repair is also possible by this BER system in eukaryotes. So what are the components needed? Let's talk about this. Some glycosylase, apart from its specificity, because glycosylases are monomer, they have no cofactors, okay, but they have epi-lyase activity. So by removing the damaged base, by cleaving the beta-N glycosidic bond, it can make a 3' nick. That is called epi-lyase activity. So whatever the epi-lyase activity happens by glycosidase or not, epi site is created that invites epi-endonuclease to make a cut at the 5' site. Okay. So after making the cut, according to the long patch or short patch, DNA polymerases are involved. DNA polymerase beta is involved in case of short patch repair. Okay. So they actually removes by lyase activity if DRPase is not present, fine, and they remove and they synthesize the new strand with respect to the mother strand and DNA ligase join them up. In case of very long patch repair related to eukaryote BER, there is another kind of enzyme is there that is called flap endonuclease, because you know, like flap, okay, because epi-endonuclease makes incision at the 5' end, a flap is made, fine. Then DNA pol delta as well as DNA pol epsilon goes and flap endonuclease FEN1 removes the flapping portion, okay. So here we can find the presence of several processivity factor PCNA, and in case of short patch repair we can find XRC1 as well. Let's talk about the nucleotide excision repair. It is a longer repair than that of the base, because in case of base excision repair, damaged base is removed. Here some nucleotide, 12 nucleotides will be removed. Let's talk about the prokaryotic system, which includes UVR ABC, that is called together as exonuclease. Okay. So what happens? Let's talk about its mode of action. Two monomer together dimer of UVRA as well as one UVRB scans throughout the DNA to find if any damage is there, like thymine-thymine dimer. Okay. It needs the ATP hydrolysis. After getting the damage, it makes a bend of 30 nucleotide bubble. Okay. It is also ATP dependent. Now after damage recognition, UVRA detaches, okay, but UVRB still remains there, joins with UVRC. UV or BC is a kind of endonuclease. Okay. So UV or B makes a nick downstream to the damage at the 4th base, and UV or C makes a cut at the upstream 5' end, that is your 7th nucleotide from the damage. So total 12 nucleotide will be excised out by UV or D DNA helicase 2 activity. Okay, and again DNA polymerase 1 keeps on synthesizing new strand, and DNA ligase will actually join them together. UV or B has single stranded binding protein. Before joining UV or C, it joins with the nick, as well as ATP is bound during the course of cutting, incision, but they are not yet hydrolyzed. Let's talk about the eukaryotic nucleotide excision repair. In case of eukaryotes, you know, if the excision repair is damaged, this excision repair is called dark repair. Why? Because in case of photoreactivation, you need the exposure to the normal light after UV exposure. So normal light propagate the dissociation of the thymine-thymine dimer by photolyase, but here we do not need any sort of exposure to the normal light. Okay, so that's why it is called dark repair. If dark repair is absent in case of eukaryote, which is responsible main for the excision repair to remove thymine-thymine dimer, because photolyase is absent, the system is prone to skin cancer. That is called xeroderma pigmentosum. So according to the disease name, the genes are named. XPA is important for recognition of the thymine-thymine dimer. XPB and D, they are attached to transcription factor 2H. Okay, so that is a transcription initiation factor related to RNA polymerase 2, because here the repair is associated with transcription, that is coupled. Fine. And then XPF, it makes a nick at the 5' site. XPG makes a nick at the 3' site. So somehow the thing is quite related to 24 to 29 base pair excision like prokaryotes. Let's talk about the mismatch repair system. So students, I have talked about the DNA damage and their repair by excision repair, by photoreactivation as well. So you can observe the damage that can be observed by the repair system. But mismatch is not damage until and unless the point mutation can be introduced to the next generation. Okay, so mismatch can be recognized by a specific system. Fine. So a time gap is there just after replication and another process. What is that? Methylation. There are two enzymes, DAM and DCM, DNA adenine methylase and DNA cytosine methylase. DAM add methyl group to adenine of GATC sequence and DCM add methyl group to CCA-GG sequence, fine, to the cytosine. This is not giving rise to mismatch bonding, fine, but that actually helps to escape from the restriction enzyme attack to the bacteria. Okay, but just before the methylation to the daughter strand, after replication a time gap is there. Fine, this condition is called hemimethylated condition. Mother strand A is methylated but daughter strand not yet. Fine, let's talk about the mode of action of mismatch repair in prokaryotes. So mismatch repair in prokaryotes may be for long patch, short patch or very short patch repair. So when it is for very long patch or long patch, they recruit mut S, mut L, mut H as well as mut U. Mut related to mutator. Okay, let's talk about their mode of action in prokaryotes. So 2 homodimer of mutase recognizes the mismatch. Fine, it binds over there making a loop by ATP hydrolysis. It recruits mut L. Mut L recruits mut H. Mut L is dimeric, okay, homodimer. And after the recruitment of mut H, which is an endonuclease, makes an incision just before C at the undermethylated daughter strand. Fine, and by formation of the loop, the mismatch and the total enzyme system come closer to the GATC site. Fine, so in this way the damage is recognized at the daughter strand. Fine, now when an incision is made, then DNA polymerase 1 will keep on lyase activity by chewing them up and synthesize the new strand with respect to the mother strand up to mismatch. Okay, so in this way mismatch repair happens. Mismatch repair in eukaryotes, we can find several differential homologs of mutase as well as mut-L. Mutase homologs are what? MSH1 to MSH5. In case of mut-L homologs, we can find MLH1 as well as PMS1 and PMS2. Defect in such mismatch in eukaryotes give rise to hereditary non-polyposis colon cancer. Okay, so mismatch must be removed, otherwise point mutation must be there. So students, I have talked about the causes and sources of DNA damage, the types of DNA damage, single-stranded, double-stranded as well, consequence of DNA damage, the damage must be repaired, the significance of the repair, the types of repair, factors needed for repair. I have talked about the single-stranded and double-stranded repair, but I have concentrated in damage reversal and removal related to single-stranded DNA repair. Thank you.